Hello, my dear students. Welcome you all to Sri Jain Rank Zone classes. I'm your mentor and your friend for the subject biology, and I will be taking the zoology part, that is animal science. My dear students, as we have discussed the oxygen hemoglobin dissociation curve in the last lecture, the last slide we discussed the Bohr effect and the Haldane effect. Now we are starting today with the same slide, the difference between the Bohr effect and the Haldane effect, just to memorize you and just to make you remember the facts. That Haldane effect is basically related to the association of oxygen and hemoglobin, and Bohr effect is basically associated with the dissociation of oxygen and hemoglobin. Haldane effect was first described by John Scott Haldane. Bohr effect was first described by Christian Bohr. Haldane effect basically occurs in the lungs at the levels of alveoli. At the levels of alveoli. Bohr effect occurs at the metabolizing sites, means at the tissue level. At the tissue level. Haldane effect describes the release of CO2. It describes the release of CO2. It describes the release of oxygen gas. Okay. Haldane effect is effective under high blood pH at the condition of alkalosis. And Bohr effect, acidosis. It is caused by the uptake of oxygen by the lungs. It is caused by the take of carbon dioxide at the metabolizing tissues. Okay. Facilitates the binding of oxygen to hemoglobin, facilitates the release of oxygen to metabolizing tissues. Now, these are the two graphs. One is the Bohr effect and the other one is the Haldane effect. You can see CO2 and H plus are affecting the affinity of hemoglobin or for oxygen. So most important thing you have to remember, that Bohr effect says what? That CO2 and the presence of H plus ions in the blood, that is the pH of the blood, are the major promising factors which affect the dissociation of hemoglobin and oxygen, right? Haldane effect tells that O2 is affecting the affinity of hemoglobin for CO2 and H plus. So they are antagonistic effects. Both are antagonistic effects. You can see for Haldane effect, if we plot a graph between partial pressure of CO2 and the amount of CO2 content of blood, then as we increase the partial pressure of CO2, as we increase the partial pressure of CO2, you can see the delivery, the CO2 delivery, or we can say the CO2 delivery, this is the CO2 delivery, lungs, because this effect, it happens at the level of alveoli, at the level of lungs. Okay, to beat the partial pressure of CO2 at the level of lungs. Okay, so when it is high, the oxygen is delivered. The oxygen is delivered. Okay, so if the partial pressure of CO2 is high, oxygen is associated. Oxygen combines with hemoglobin to form oxyhemoglobin. Okay, this is Bohr's effect. The graph, it is a sigmoid shaped curve. Okay, everything is discussed in the previous slide, in the previous video. Okay, I will not take your time to describe it again because everything is understood. Okay, this is important. Lungs and tissue, at the levels of lungs and tissue. So at the level of lungs, you can see the dissolved O2, HbO2, oxyhemoglobin 97%, 3%. Okay, CO2 mainly travels in the form of dissolved form. Okay, and very less amount of CO2 travels in the form of carbon immunohemoglobin, that is with the RBC, in the with the uptake of RBC. Okay, so the CO2 transport we have to start today. Okay, so you can see the HbO2 combines with H plus to form reduced hemoglobin and releases oxygen gas. This oxygen gas is taken up by the tissues. Okay, and the HbO2 combines with CO2. Okay, to form acid. This you can see the hemoglobinic acid. This is hemoglobinic acid or hemonamic acid. Okay, plus H plus plus O2. So these two reactions in which the reduced hemoglobin is formed, or we can say the hemoglobinic acid is formed, they ultimately increases the production of CO2. Okay, reduced hemoglobin and hemoglobinic acid or hemimic acid. Okay, now transport of CO2, we have to start with transport of CO2. In dissolved state, because of high solubility of CO2 gas, around 7% of carbon dioxide gets dissolved in the blood plasma and is carried in solution to lungs. It is carried in solution to lungs. Deoxygenated or venous blood and oxygenated or arterial blood carry about 2.7 ml and 2.4 ml of CO2 per 100 ml of blood in dissolved state in plasma respectively. Okay. In dissolved state, you can see the CO2 gas is much more soluble in comparison to oxygen. Okay. Around 7% of carbon dioxide gets dissolved in blood plasma and is carried in solution to lungs. Deoxygenated or venous blood and oxygenated or arterial blood carry about 2.7 ml and 2.4 ml per 100 ml of blood respectively, the amount of CO2 in dissolved form. Okay. The dissolved form or the dissolved carbon dioxide in blood reacts with water. It reacts with water to form carbonic acid H2CO3. H2O combines with CO2. The water from plasma combines with CO2 gas, carbonic acid. The reaction is reversible and somewhat non-spontaneous. So a carbonic anhydrase enzyme makes the reaction much more spontaneous. Okay. This reaction is very slow in blood plasma but occurs very rapidly inside RBC because zinc containing enzyme carbonic anhydrase, very important, present in RBC, accelerates its rate around 5,000 times. So the dissolved carbon dioxide in blood reacts with water to form carbonic acid. The reaction is very slow in blood plasma but occurs very rapidly in the presence of a zinc containing enzyme, that is carbonic anhydrase, which is present in RBC. So inside RBC, the same reaction travels 5,000 times faster than the plasma. Due to this, about 70% of CO2 received by blood from the tissues enters the red blood corpuscles where it reacts with water to form carbonic acid H2CO. Due to this, about 70% of carbon dioxide received by blood, 70% of CO2, which is carbon dioxide, which is received by blood from the tissues, enters the RBCs where it reacts with water to form carbonic acid, that is H2CO3. Please rectify this term. It is not about 7%, it is 70% of CO2 gets dissolved in the blood plasma and is carried in the solution to the lungs. Okay. Yeah. Yeah. This is 70%. Okay. Carbonic anhydrase is exclusively found in red blood corpuscles. All the other tissue contains it in traces except stomach and pancreas, in which have it is having or it is present in considerable amount. This enzyme not only speeds up the formation of carbonic acid H2CO3 but also rapidly convert it back to carbon dioxide and water when blood reaches the lungs. Okay. So the carbonic anhydrase is exclusively found in RBCs. All the other tissue contains it in traces except stomach and pancreas where it is present in considerable amount. This enzyme not only speeds up the formation of carbonic acid but also rapidly converts it back to CO2 and water when the blood reaches the lungs. Almost as rapidly as formed, all carbonic acid of RBC dissociates into H plus and bicarbonate ions like this. H2O combines with CO2 to form H2CO3, which further dissociates to form H plus HCO3 minus. The most of the bicarbonate ions formed with RBCs diffuse out into the blood plasma. It comes out into the blood plasma along the concentration gradient. Okay, so this is RBC. Suppose I'll make the picture. This is RBC. Okay, in RBC, H2O combines with CO2 to form H2CO3, which further forms H plus and HCO3 minus. This HCO3 minus comes out from RBC along the concentration gradient. When the whole blood is saturated with carbon dioxide, the following changes are seen. When the whole blood is saturated with CO2, then the following changes are seen. The bicarbonate content of plasma and corpuscles increases. Bicarbonate content of plasma and corpuscles increases. The chloride content of plasma is diminished and that of cells is increased. The bicarbonate content of plasma and corpuscles increases, both in plasma and corpuscles. The chloride content of plasma, because Cl minus comes out, the chloride content of plasma uh, diminished, okay, and that of cells is increased. The total base means the cations of both plasma and corpuscles remains unchanged, the condition becomes electrically neutral, and the water content and the volume of corpuscles increases. So the bicarbonate content of plasma and corpuscles increase, the chloride content of plasma is diminished and that of cells is increased. The total base or the cations of both plasma and corpuscles remains unchanged. The water content and volume of corpuscles increases. Water content of corpuscles increases, volume of corpuscles increases. Okay? As the HCO3 minus ions comes out to balance, Cl minus comes in. So Cl minus increases in the corpuscles, HCO3 minus decreases in the corpuscles. Okay, but the HCO3 minus formation is so fast that that gap is already maintained, and both in corpuscles and plasma, HCO3 minus increased. So you have to remember HCO3 minus increased both in plasma and RBC. Cl minus increase in RBC, decrease in plasma. Okay. Total base or the cations of both plasma and corpuscles remains unchanged. The water content and the volume of corpuscles increases. When the CO2 is removed from a sample of blood, reverse change takes place. When CO2 is removed from the sample of the blood, reverse change takes place. From these observations, it is evident that when CO2 enters the blood, chlorine from plasma enters the RBC. When CO2 enters the blood, CO2 comes out, chlorine from the plasma enters the RBC while the base is left behind. Okay, base is Na. Okay, Na. How H2O combines with CO2 to form H2CO3, carbonic acid, to form H plus plus HCO3 minus. This H plus combines with hemoglobin to form reduced hemoglobin HHB. NaHCO3 comes out of RBC to plasma to form NaHCO3. Okay, NaHCO3 is formed because NaCl combines with HCO3 minus. Okay, out of RBC to form plasma to form NaHCO3. So NaCl combines with HCO3 minus to form NaHCO3, and Cl minus comes in. It goes in. Okay, and the Na plus is left behind. When CO2 escapes the plasma and combines with base again due to this alternate movement of chlorine ions, this phenomenon is called chloride shift or Hamburger's chloride phenomenon. It is known as what? Hamburger's chloride phenomenon. When CO2 is removed from a sample of blood, reverse change takes place. From these observations, it is evident that when CO2 enters the blood, okay, chlorine from plasma enters the RBC while the base is left behind. This base combines with CO2 again to form bicarbonate ions, okay, and again the Cl minus, uh, it comes out and then comes in. So this alternate movement of chlorine ions is known as what? Hamburger's chloride shift or chloride phenomenon. You can see the CO2 plasma, red blood cell, H2CO3. H2CO3 forms KHB. Okay, KHB simply is what? That potassium is present in the hemoglobin. This potassium combines with HCO3 minus. This potassium combines with uh, the CO2 or H2CO3 to form KFCO3, potassium bicarbonate, and reduced hemoglobin is formed. Reduced hemoglobin is formed. This reduced hemoglobin. Now, the Cl from dissociation of NaCl, Na plus and Cl minus. So Cl minus combines with KHCO3 to form HCO3 minus ions, and KCl is formed. HCO3 minus ions comes out. Okay, this is red blood cell, this is plasma. This is RBC, this is plasma. So Cl minus, it goes in from plasma to RBC. Okay, and SCO3 minus ions, it comes out. This HCO3, KCl, and HHB, they all increase in red blood cell. So HCO3 minus increases both in red blood cell and plasma. Cl minus increases in red blood cell. HHB also increases in red blood cell. Understood? Okay, this is the buffering action of HCO3 minus. Act as a buffer. It maintains the pH. Okay. Now, this is the Hamburger's chloride shift or chloride phenomenon as carbaminohemoglobin. In addition to reacting with water, CO2 also reacts directly with amine radicals, NH2 radicals of hemoglobin to form unstable compound carbaminohemoglobin. This is a reversible reaction. Okay. So NH2 moieties which are present in the hemoglobin molecule, in addition to reacting with water, carbon dioxide also reacts with this NH2 or amine radicals of hemoglobin to form unstable compound carbaminohemoglobin. This is a reversible reaction. A small amount of CO2 also reacts in this same way with the plasma proteins. A small amount of CO2 also reacts in this same way with the plasma proteins. About 23% CO2 is transported in combination with hemoglobin and plasma proteins. Okay, so this is reduced hemoglobin, NH2 moieties. It combines with the CO2 to form reduced hemoglobin. This is reduced hemoglobin combines with CO2 to form carbaminohemoglobin, okay, along with the removal of H plus. So in addition to reacting with water, carbon dioxide also reacts directly with the amine radicals of hemoglobin to form an unstable compound, carbaminohemoglobin. This is a reversible reaction. A small amount of CO2 also reacts in the same way with the plasma proteins, okay. About 23% CO2 is transported in combination with hemoglobin and plasma proteins. Hemoglobin as a buffer. Addition of hydrogen ions would make the blood acidic. However, most of the hydrogen ions are neutralized by combination with hemoglobin, which is negatively charged, forming acid hemoglobin or hemoglobinic acid. This reduces the acidity of blood and also releases additional oxygen. Addition of hydrogen ions would make the blood acidic. However, most of the hydrogen ions are neutralized by combination with hemoglobin, which is negatively charged, forming acidic hemoglobin. This reduces the acidity of blood and also releases the additional oxygen. If the blood becomes too basic, acid hemoglobin dissociates, releasing hydrogen ions. Okay, H plus ions are released. Thus, the hemoglobin also acts as a buffer, a substance that keeps the pH from fluctuating. HHB to form H plus plus hemoglobin. Okay, the hemoglobin of the fetus has higher affinity for oxygen than the mother's hemoglobin. After birth, the fetal hemoglobin is gradually replaced by adult hemoglobin. The fetal hemoglobin is much more affinitive towards oxygen than the mother's hemoglobin, and after birth, the fetal hemoglobin is gradually replaced by adult hemoglobin. Okay, so when the hemoglobin, it acts as a buffer as well, because reduced hemoglobin, it can release H plus, it can donate H plus. Okay, the hemoglobin act as a buffer along with HCO3 minus ions. They also act as a buffer. Okay, they help the exchange of HCO3 minus with Cl minus. Okay, to maintain the electrical neutrality is known as what Hamburger's chloride phenomena you have just studied in the previous slide. What are the respiratory pigments and the different animals which are containing different respiratory pigments? Hemocyanin. Hemocyanin is copper based, copper containing blue pigment because of Cu2 plus in plasma of crustaceans, snails, and cephalopods. So hemocyanin is a respiratory pigment, okay, containing copper. It is a blue pigment occurring in plasma of crustaceans, snails, and cephalopods. Chlorocurinin. Chlorocurinin is an iron-containing green pigment occurring in plasma of annelids and polychaetae. Hemocyanin is present in arthropods, uh, uh, like crustaceans, snails, and cephalopods. Okay, so it is present in crustaceans, snails, and cephalopods. Chlorocurinin, it is an iron-based pigment which is present in the plasma of annelids and polychaetae. Pinaglobin. Pinaglobin, it is a manganese-containing brown pigment occurs in the body fluid of some mollusca. Pinna. Okay. So it is the manganese-containing brown pigment occurs in the body fluid of some mollusk like echinochrome contains iron and occur in the coelomic fluid of sea urchin. Echinochrome contains iron and occur in the coelomic fluid of sea urchin. Iron and occurs in the coelomic fluid of sea urchin or echinoderms. Okay. So echinochrome, chlorocurinin, pinaglobin. Okay. Now, vanadium contains vanadium present in the blood of tunicates, Urochordata, Cyona, Cliona contains vanadium in plasma. Ascidia contains green blood corpuscles, vanadidocytes. Okay. So, Cliona contains vanadium in plasma and Ascidia contains green blood corpuscles, vanadidocytes. Myoglobin, hemoglobin of the muscle, molapidin occurs in Molapedia, echinodermata. Okay. These all are different, especially you have to remember the myoglobin, vanadium, it's important, hemocyanin, chlorocurinin. Okay, these are some important ones. Okay, now the neural control, we are going to start with the neural control and the chemical control. The respiratory system, basically it is bifurcated into two major types: the neural and chemical control. It controls the rate of respiration. Respiratory center is located in medulla oblongata and pons varolai. It has following well dispersed components. Okay, it controls the rate of respiration. Respiratory center is located in the medulla oblongata and pons varolai. It has the following well dispersed components. First is the dorsal respiratory group. Okay, located dorsally along the length of medulla with neurons interconnected to sensory termination of glossopharyngeal nerve. Okay, you know glossopharyngeal is a cranial nerve. So it controls the rate of respiration, and the respiratory center is located in medulla oblongata and pons varolai. It has the following well dispersed components. One is the dorsal respiratory group. It is located dorsally along the length of the medulla with neurons interconnected to sensory termination of glossopharyngeal sensory signals from peripheral chemoreceptors. Chemoreceptors, they are peripheral, central, and inferior, and vagus nerve, sensory signals from lungs and stretch receptors of bronchi nerves. So dorsal respiratory group basically located dorsally along the length of the medulla with neurons. It is having neurons which are interconnected to the sensory termination of glossopharyngeal nerve and the vagus nerve. So sensory termination of glossopharyngeal nerve brings the sensation from peripheral chemoreceptors, and vagus nerve, the vagus nerve, it carries the signals from lungs and stretch receptors of bronchi. Okay. The area is connected through nerves to phrenic muscles of diaphragm. Nervous signals from this group brings about the normal resting inspiration. Expiration occurs through elastic recoil of thoracic wall and lungs. Basically, we have a dorsal respiratory group which controls the normal resting inspiration and the recoil capacity of thoracic wall and lungs for expiration. Okay, the area is connected through nerves to phrenic muscles. The area is basically connected to nerves to phrenic muscles. Through nerves to phrenic muscles means the muscles which are supplied to diaphragm. Okay, neural control of respiration. You can see these are the respiratory groups: the ventral respiratory group, the pontine respiratory group, which includes pneumotaxic center and aneustic center, Dorsal respiratory group. Dorsal respiratory group you have just studied. Plus is for stimulation and negative is for inhibition. Okay. So the dorsal respiratory group, it basically stimulates the elevation of ribs by external intercostal muscles and the diaphragm, that is phrenic muscles. Okay. Internal intercostal muscles are basically stimulated by ventral respiratory group. Okay, or the accessory respiratory muscles are also activated by ventral respiratory group. Okay, pneumotaxic area. So after dorsal respiratory group, we are going to discuss about pneumotaxic area. It occurs in ponds and is meant for switching off normal inspiration when the limit of lung fill is reached. The latter is, however, also dependent upon the strength of signal, which is around 0.5 per second when the signal is strong and 5 seconds when signal is poor. Pneumotaxic area, it occurs in ponds, is meant for switching off normal inspiration, normal breathing, okay, when the limit of lung fill is reached, okay, as the lungs come up to their maximum capacity to hold the air, okay, to fill up with the air, the pneumotaxic area switch off the normal inspiration process. The latter is, however, also dependent upon the strength of signal, that is 0.5 second when signal is strong and 5 second when signal is poor. Then ventral respiratory group VRG, it occurs ventrolaterally anterior to the dorsal respiratory group. The group has two types of neurons, inspiratory and expiratory. They are normally inactive, but when the respiratory drive is greater than normal, the group is activated. It results in deeper and quicker inspiration and expiration. So deep inspiration and expiration is basically controlled by ventral respiratory group. It occurs ventrolaterally anterior to dorsal respiratory group. The group has two types of neurons. One is the inspiratory and the other one is expiratory. They are normally inactive, but when the respiratory drive is greater than normal, the group is activated. It results in deeper and quicker inspiration and expiration. Chemosensitive area. It lies in the medulla near the place of entry of glossopharyngeal and vagus nerves. It is sensitive to blood carbon dioxide and hydrogen ion concentration. Chemosensitive areas are connected to other areas of respiratory center. It lies in the medulla near the place of entry of glossopharyngeal and vagus nerve. In a medulla where the glossopharyngeal and the vagus nerves enter. So at the point of entry, chemosensitive area is present. It is sensitive to blood CO2 and hydrogen ion concentrations. Chemosensitive areas are connected to other areas of respiratory center. So these are chemoreceptors. They contain mainly chemoreceptors. Here are two types of receptors. One is the chemoreceptor, the other one are baroreceptors. Baroreceptors get stimulated by stretch of epithelium because of pressure. Okay. The higher centers of the brain control speech, emotions, voluntary control of breathing, action potentials, and motor pathways. Okay. Medulla is the respiratory center. Medulla chemoreceptors, decrease in pH, increase in CO2, deep breathing. Input to respiratory centers in the medulla oblongata and pons modifies respiration. Carotid and aortic body chemoreceptors, okay, because of high oxygen. Herring-Beer's reflex or stretch receptors in lungs, they stretch to fill in maximally, okay. So stretch receptors inhibit the, uh, rate of respiration. Proprioceptors in muscles and joints stimulates the rate of respiration. Receptors for touch, temperature, and pain stimuli also gives direct input to his respiratory centers in the medulla oblongata and pulse for the modification of respiration. So higher centers of brain, medulla chemoreceptors, carotid and aortic body chemoreceptors, Herring-bearer reflex, stretch receptors in the lungs, proprioception in muscles and joints, they all have their impact. Okay, like we can say the higher receptors of the brain, okay, they can perform dual function. Okay, both the inhibition as well as the stimulation. Okay, herring-bearer reflex inhibits the uh, rate of respiration. Respiratory disorders we will be starting in the next turn. Okay, please be very sure that all these, especially the neural control and the chemical control because of decrease in pH or high amount of CO2 and neural control, these all concepts should be very through, because the questions we will discuss in the coming class, they will be maximally of the same category, they will belonging to the same topic, that what are the different regulatory mechanisms of respiration, how the rate of respiration is maintained. So we will be talking about respiratory disorders in the next turn. Till then, bye bye. Take care. Stay safe. Stay healthy.